The importance of the gut-brain axis in regulating stress-related responses has long been appreciated. More recently, the microbiota has emerged as a key player in the control of this axis, especially during conditions of stress provoked by real or perceived homeostatic challenges. The gut microbiome is made up of all the microbiota that live in our intestinal tracts, including the stomach. These minuscule organisms, especially bacteria, help the body break down carbohydrates, proteins, and sugars into useful nutrients and process fiber in the colon. The roots of communication between the microbiota and brain are slowly being unraveled, and include the vagus nerve, gut hormone signaling, the immune system, tryptophan metabolism, and microbial metabolites such as short-chain fatty acids. The gut microbiota has been implicated in a variety of stress-related conditions including anxiety, depression, and irritable bowel syndrome, IBS. Additional research in humans is much needed to reveal the relative impact and causal contribution of the microbiome to stress-related disorders. In this regard, the concept of psychobiotics is being developed and refined to encompass methods of targeting the microbiota in order to positively impact mental health outcomes. The microbiome also affects the structure and function of the amygdala, another key stress-related brain region. The amygdala is critical for emotional learning and social behavior, and is critical for the gating of behavioral and physiological responses to stressful stimuli, especially those that trigger anxiety and/or fear. The precise role played by the microbiota in gut-brain signaling pathways remains to be understood. Hindering our efforts is the limited current state of knowledge regarding the identity and function of the gut's vast and diverse microbial composition. A complex communication network exists between the gut and the central nervous system, CNS, which includes the enteric nervous system, ENS, sympathetic and parasympathetic branches of the autonomic nervous system, ANS, neuroendocrine signaling pathways, and neuroimmune systems. Serotonin, 5-HT, is a biogenic amine that functions as a neurotransmitter within the brain and also within the ENS. Indeed, approximately 95% of 5-HT within the body is produced by the gut. One of the principal mechanisms proposed to underlie a gut-brain link in depression and stress-related disorders is via disrupted gut barrier function, commonly known as the leaky gut phenomenon, which is proposed to contribute to major depressive disorder, MDD. The proposed mechanism of action is that the epithelial barrier of the GI tract becomes compromised as a result of psychological or organic stress, leading to increased intestinal permeability and subsequent translocation of gram-negative bacteria across the mucosal lining to access immune cells and the ENS, producing an inflammatory response. Major depressive disorder, MDD, is associated with changes in gut permeability and microbiota composition. Anxiety and depressive disorders may be characterized by a higher abundance of pro-inflammatory microbiota and less short-chain, fatty acid-producing bacteria. Episodes of anxiety and depression may be experienced more frequently in patients with GI disorders like irritable bowel syndrome, IBS. Negative emotions, stressful life events, and personality traits like neuroticism have also been associated with colitis, Crohn's disease, and dyspepsia. Several groups have documented the beneficial effects of probiotics and prebiotics in healthy individuals. Indeed, the idea that lactobacillus strains may improve quality of life and mental health is not new. Diet is one of the most important modifying factors of the microbiota gut-brain axis. Diet functions as a major pathway from stress to gut dysbiosis. Even mild stressors can encourage unhealthy eating. Stress and depression can also alter metabolic responses to food. Diet has emerged as one of the most powerful predictors of gut bacteria composition, above and beyond one's genotype. Diet determines which bacteria will thrive in the gut, and the gut bacteria in turn aid digestion. The Western diet, high in saturated fat, processed foods, and refined sugar, starkly contrasts with fiber-rich, plant-based diets of indigenous cultures. The Western diet fosters a distinct gut microbiota signature with low gut microbiota diversity and greater gut leakiness, which may contribute to metabolic syndrome and chronic disease onset. In particular, imbalanced macronutrient intake may be to blame. For instance, low fiber consumption may dysregulate immune function, as short-chain fatty acids, SCFAs, resulting from bacterial fermentation of complex carbohydrates are important for healthy immune function. Likely due in part to the Western diet, 60 to 70 million Americans suffer from digestive disorders, costing $100 billion annually. Importantly, some digestive medications impact the gut bacteria. For example, Proton pump inhibitors, PPIs, e.g. omeprazole, widely used to treat acid reflux, reduce diversity and affect 20% of species, creating an unhealthy gut microbiota that may predispose to gastrointestinal infection. Indeed, PPIs may negatively impact the gut bacteria population even more so than do antibiotics. In conclusion, researchers are furthering their understanding of the delicate connections between the human gastrointestinal, GI, microbiota and the brain. The bidirectional communication along the gut-brain axis involves many organ systems, including the endocrine system, the immune system, and the autonomic, central, and enteric nervous systems, with the intestinal microbiota influencing these interactions. The enteric nervous system, ENS, is able to operate GI function independently of the central nervous system, CNS. However, many of the pathways and neurotransmitters are similar between the nervous systems in the brain and the gut. It is now understood that diet plays a significant role in shaping the microbiome, with experiments showing that dietary alterations can induce large, temporary microbial shifts within 24 hours. For example, it has been shown that a high-fat diet adversely reduces beneficial bacteria. Consuming fiber, for instance, can increase microbiota diversity, reduce blood glucose levels, and help people maintain a healthy weight. Therefore, there can be significant therapeutic utility in altering microbial composition through diet. Pre- and probiotics can be very helpful in aiding gut flora diversity. Additionally, newer studies show that the role of regular exercise, which improves a wide variety of metabolic functions, including stress reduction, has also been shown to have salutary effects on the gut microbiota. Managing external stress-related factors while optimizing gut health may jointly address some chronic health conditions. Specifically, personalized therapeutic strategies that combine stress transformation approaches with gut health interventions, such as nutritional therapies, supplements, and nutraceuticals, may help to optimize gut function and bolster related body systems. Stress, prolonged and chronic, is dangerously unhealthy. There are many strategies that can aid in managing stress and improving quality of life. My video on my YouTube channel, Zen in the Chaos, Strategies for Stress Management, offers guidance on stress management techniques and behaviors. My website, besthealthwithlee.com, is your gateway to high-quality, life-changing diet supplementation at wholesale member prices.